And now I'd like to turn it over to our first speaker, Nathan Schlupen.
Yeah, thanks for that intro. And again, welcome to you all, and thanks for joining. So I'll quickly run you through our agenda for today and give a brief intro to the speakers of today. Then I'll hand over to Joanna to give you a general introduction on automated dissolution testing. Then I will take over again to present the results of a survey on automated dissolution testing we've done some time ago. And then we'll wrap up with a Q&A that will be moderated by parts, and we will all participate to try and answer any questions you might have. So please indeed pose your questions in the Q&A feature so we can tackle them at the end. So next slide, please. So yeah, it's not so polite, but since I'm first, I will start by introducing myself first. So I'm Nathan Schulpen. I have a master's in chemistry, and I've been working at Johnson & Johnson for the last 10 years in the Dissolution Sciences group, where we do both biorelevant dissolution testing to characterize drug products during development and also develop QC methods across the stages of development. More recently, I've actually transitioned to a broader role within AD, but still focusing on automation and new technologies across techniques, and still heavily involved in automated dissolution testing as well. The next speaker will be Johanna. Johanna works at Boehringer Ingelheim as a principal scientist. She has a Master of Science and PhD from the Kiel University. She's also over 10 years of experience in analytical development, focusing on dissolution specifically, and she's currently responsible for QC dissolution method development and validation, automation, as well as leading biorelevant dissolution activities for solid oral dosage forms. Next, we have Parth Kumar Patel, who is a pharmaceutical scientist with over 13 years of experience in CMC development, biopharmaceutics, and dissolution. He's currently a principal scientist and leads the dissolution expert group at UCB Pharma, where he drives strategic dissolution and drug product analytical initiatives and contributes to global regulatory submissions. Prior to joining UCB, Parth also held key biopharmaceutics and dissolution roles at Novartis and Intas Pharmaceutics, where he supported method development, modeling and simulations, and early to late stage development. And finally, we have Amjad Mahmood joining us at the end for the Q&A session. He has a Bachelor of Science in Chemical and Pharmaceutical Sciences, and is a principal scientist at GlaxoSmithKline in the UK, with over 15 years of experience in pharmaceutical development, specializing in sample preparation and dissolution automation of oral solid dosage forms. So with that, I would like to hand over to Johanna for an intro on automated dissolution testing.
Thank you, and a warm welcome also from my side, and thank you for taking time in your busy schedules to attend our webinar today. In the next couple of minutes, I'd like to introduce you to automation in dissolution and also define some terms which are then important for the survey and the following discussion. In general, any kind of dissolution device can be automated, so being it your USP1 to USP7 or whatever you have in your lab, or even your self-built small-scale biorelevant dissolution setups. But for the context of today's webinar and also our survey, we will focus mostly on USP apparatus 1 and 2, as those are the most frequently used systems during development and quality control for solid oral dosage forms. So in general, as I guess most of you already know, dissolution testing is a performance test, which is used throughout development and also evolves depending on the stage of your development or formulation design. It is adapted throughout development with respect to the formulation design at that stage and also dosage strength. Next slide, please. From a regulatory perspective, we have several pharmacopoeias referencing the solution. Most of them have been harmonized across USP, European Pharmacopoeia, and, for example, Japanese Pharmacopoeia, and in part the Chinese Pharmacopoeia, or ICH. And when they talk about the solution, they just talk basically about the vessel in which we run our test, and they have defined how it looks like with size and how the curvature of the bottom, for example, is. They have defined what the pedal looks like, or the basket apparatus, and they also mostly emphasize reproducibility. With that in mind, that usually refers to manual sampling as the gold standard, because that is something everybody can do if they have the vessel and the basic setup. Doing sampling, usually for manual sampling, a cannula or any kind of sampling device is introduced into the vessel. Then a sample is drawn, which then needs to be filtrated or centrifuged right away to avoid later on dissolution of previously undissolved larger particles. And with that comes also the responsibility to think about if filtration has any kind of effects on your dissolution setup. But that's basically it for manual dissolution. Next slide, please. When we now talk about semi-automated dissolution, we introduce a lot more variables into the system. So you have a kind of sampling device which draws samples from your dissolution setup. Usually this is introduced into the vessel during the predefined sampling times. And then we need to think about filters. So we can either have a cannula filter, which is added to the bottom, which is also introduced into the vessel, or you can have other filters right after drawing the sample. Depending on how much automation you use, you can also have a filter station which changes your filter for you, or you just use one filter throughout the entire setup. What is the largest new part is the introduction of tubing when we talk about automation. So when we draw the sample, it is not drawn in a syringe, which we do manually, and then fill it into, for example, HPLC vials. But we now have a pump pumping your samples through tubing, either into an autosampler, which collects it into HPLC vials or test tubes, or you can have online detection via HPLC or photometer. And for the photometer, you can also have a closed loop system, which basically pumps back your volume. So in our context for this webinar and the survey, we defined a semi-automated dissolution system as a system which is capable of automated sample collection and filtration and or online analysis. And if you have, you can also have an automated filter device or automated sample introduction. In that context, often terms like online analysis and in situ analysis come up. So we define them as online analysis is if the sample is analyzed concurrent with the dissolution run outside the system. So if you look at this graph here, that would mean the sample is pumped through the tubing to the photometer and maybe back again and analyzed right beside your dissolution system. So it's sort of in time, but a couple of seconds later then it would be in the vessel. And in contrast, in situ analysis is the analysis directly inside the dissolution vessel, which can be done, for example, by introducing a UV fiber probe, which is then put directly into the vessel and basically omits the use of or the need for filters or tubing. But on the other hand, it introduces potential some kind of disturbance within the vessel. Click, please. And because we now have tubing, we also need to worry about what we pump through that tubing. So when we draw a sample, we need to take the length of the tubing and the volume going through it into account, which needs to be pumped through the autosampler or the online detector. And by doing so, you also need to take into account that when you draw a sample at a predefined time, you need to start pumping earlier to have the sample drawn at the correct time in your vessel, in your HPLC vial or in your photometer at the correct time. Also depending if you have at absorption effects to your tubing, for example, you may need to increase the volume to pump through the tubing. With the prime volume, we mean the volume which is pumped prior measuring or sampling your samples through the tubing, prior, and with the waste drop volume, that can be like instead of pumping it back into your system, you can also discard the volume into the waste, and basically that is the amount which you need to pump prior filling your HPLC vial. Next slide, please. So here are some examples for semi-automated systems and different variation of automation. And I try to be as inclusive as possible. So those are like from all different vendors, and every vendor has their own setup and preferences on how they do automation. So on the upper left-hand part, we do have a system where we have a dissolution tester just combined with an auto sampler. So that can draw samples into HPLC vials or test tubes. On the upper left-hand part, we do have a dissolution system coupled with an auto sampler, which also serves as a pump into a photometer, and it can either do online analysis and or collect samples. On the lower left-hand part, we have a system with a dissolution tester, a auto sampler, and a volume replacement unit. So depending on how much sample you draw, and sometimes that can be quite significant. If you have absorption effects, for example, you may need to do filter media replacement, and that's what the system can do. And on the lower right-hand side, you have a system coupled with a in situ analysis tool, which is like a UV probe in that case. That increases the amount of time points you can measure significantly, as you do not need to draw samples and pump them to a detector or for analysis. It also can omit the need for filters. However, those in situ devices have different shapes, and depending on the shape you choose, they can introduce some turbulences into your vessel. So that needs to be checked if that has an effect on the samples. Next slide, please. So in contrast, basically the fully automated system does everything for you. So the semi-automated system, depending on how much automation you want, you can have a little bit or a little bit more. And the fully automated system is a system which can do the entire setup for you, like automated vessel filling, automated sample introduction, automated sample collection and filtration, and or online analysis, as well as automated vessel and system cleaning. This is necessary because you want to run multiple runs after each other, and thereby you also need to think about preloading the samples into the system. Click, please. Those fully automated systems are often capable of preparing the media for you, either from barrels where you have 50 or 100 liters medium prepared. They can preheat them and degas them, or you can even do media preparation from concentrates, where the automate does mix the concentrates with water prior preheating and degassing. The system can automatically clean themselves. However, that also introduces some limitations. For example, it's on filter. So cannula filters are usually not suitable for use in fully automated systems, and even the syringe filters, for example, sometimes need predefined casings, otherwise they don't work with the filter stations. Also, with fully automated dissolution, you create a lot of data, so IT interfaces are crucial to manage the data and put them directly into your system. Next slide, please. So those are two examples for fully automated dissolution. On the left-hand side, this is a dissolution bus connected with, like the white part in the middle is a automated filter station, and then a auto sampler which is also connected to a photometer. On the right-hand side, it's basically the same system, but from a different vendor, which has also a video capability, so you can monitor your dissolution release in every vessel during every run, and it can also be coupled with HPLC. Next slide, please. So what are the challenges? So basically everything is a challenge, which is different to manual sampling. So overall, you need to make sure that everything in your system is compatible with each other. So while you can choose different manufacturers for your different parts, they may not be compatible software-wise, or you need to be creative or have in-house software solution for that. To omit that, you basically need to purchase your instrument from the same vendor or all the parts from the same vendor. Often transfers from one site or one lab to another lab are done manually because it's easier if everybody just draws a manual sample from the vessel and omits all the different automation steps. As not all sites may have the same automated equipment. So you need to make sure that your manual method is similar to your automated methods and not a lot of artifacts are introduced. And by introducing so many parameters, qualification of new systems and validation in GMP environments become a little bit more challenging. Qualification, especially also because you need to worry about the software and if that works in your infrastructure and systems. And validation because you usually add additional robustness parameters, which would not be needed in manual methods. And as I said before, the infrastructure is key as you need interfaces for data management and also starting or managing the experiments. Next slide, please. So why would you do automation? I guess the largest benefit is that you can increase throughput if you have a function method. Fully automated systems can run through the night or through weekends and holidays. Semi-automated systems, basically, if they work well, they just need to be cleaned in between. But overall, everything else runs by themselves or a lot more smoothly. You omit or reduce human error, especially if you have samples which are sensitive to any kind of disturbance in the vessel, and you have different people drawing samples and maybe pushing more or less with less force the sample into the vessel can have an impact. And by that, you overall improve accuracy and precision. And I guess safety and health of workers would also be a huge point, especially in early development when APIs are not fully characterized and you don't know how dangerous they are. This can reduce the context of the people working in the lab with your compounds quite a bit. The cons are the initially high cost. So in general, compared to just buying a standard dissolution device, fully automated systems are at least 10 times more expensive or even more. And it's not just the device because you also need the correct infrastructure, which means you need access to the correct water and also waste disposal because you would run automatically everything through your system. And also the IT infrastructure is really, really important, not just with respect to the software run on the system, but also to connect them to your system, for example, or to Empower. Depending on the system, they have limitations in terms of which filters you can use, or if you have higher amounts of surfactants, which lead to foaming, that might not be possible if there's a little bit of air introduced during the dissolution run. And yeah, overall, the complex qualification and validation may become a hindrance. And depending if you have more than one system, you introduce a lot more software, which you need to train your technicians and also keep up to date in the ever-evolving software environment we have now. So with that, I will hand over to Nathan to show you our results from the survey.
Yeah, thank you, Johanna, for that introduction. Indeed, so please, you can move to the next slide. To start, maybe some context on the survey we did. So we sent out a survey on automated dissolution some time ago that was distributed to the members of the IQ Consortium. And we really wanted to focus on application of automated dissolution testing, general practices, and common hurdles. And then with that data, our goal was to organize a webinar. So that's what we're doing today. Following that, we also intend to prepare a publication on the use of automated dissolution testing and common issues. And then finally, we also just wanted to get a sense of how widely spread is automated dissolution testing today, and use the info we gained from the survey as input for our sub-team to define the mission and understand where we could make a potential impact. Next slide, please. So for starters, we had quite a good response. We had 18 responding companies. That doesn't mean it's 18 individual responding. There might be multiple users within the same company, and the companies just submitted a consolidated answer. Within those companies, there might be actually different labs or users using automated systems. We did also ask for people not using automated dissolution testing to answer to the survey. They could then obviously only answer the first question, meaning, no, we don't use automated dissolution testing. We didn't get any one responding that, so we're not sure whether people who didn't use automated dissolution testing did not respond, or whether just everyone who saw the survey and participated is using automation in dissolution. When we look then at the level of automation people use, that's the left graph, we see that actually even the majority of companies responding they use automated dissolution testing use both semi and fully automated systems, close to 3/4, and only 1/4 more or less uses only semi-automated systems, which was in a way kind of a positive surprise to us. I would say that even the use of fully automated system seems to be quite widespread in the industry. Then if we look at the right graph, that's where we actually asked responders where they applied or in which phases they apply automation for dissolution testing. And please be careful here, because this is not expressed in percent, but in number of respondents. So the maximum is 18, basically. When we look at the use of automation for semi-automated systems, we see that all responders basically use semi-automated dissolution testing in R&D phases. And then also still majority use it during clinical development phases. And also in a lot of responders use semi-automated dissolution testing during the commercial phase of the product. Looking at fully automated dissolution testing, keeping in mind that only, let's say, 13 responders, I think, have fully automated systems. Also, close to all responders actually use their fully automated system in an R&D setting. And here the trend is a bit different, that it's used a bit less in clinical settings, but actually more in a commercial setting. So potentially there's even companies using it more towards commercial than for R&D or clinical. So you can move to the next slide, please. So then we asked some questions more focusing on the features of the automated system. And this first question contains quite a lot. So basically we asked, what type of features does your automated system have? So maybe starting with semi-automated system on the left, we asked for three features. And one is a filter changer. Johanna alluded to it before. You can either change the filter or not change the filter during the dissolution test. But if you don't have an automated filter changer, you will not be able to change it. And then we see 3/4 of participants, they actually have a filter changer in their semi-automated system, meaning 1/4 doesn't. Which is quite an interesting result still, I would say. And then if we look at the use of online UV, or the availability of online UV capabilities with the semi-automated systems, we see that 80% of respondents actually have online UV coupled to their semi-automated system. There's quite some reasons why you would do that. Again, that this is quite a high number, so it's quite widespread. And of course, if you have online UV measurements, this means you will have your data available real time. There is still limitations, of course, to UV testing. And that's why you also see that 20% of people actually have online LC capabilities, meaning you can couple your semi-automated system directly to an LC, so it can directly inject the samples you have taken. Depending on the technology you use, it might still be collected in vials first or not. And then you still have the benefit basically of having the chromatography and the separation, but still you have the online capability. And then if we look at the features, sorry, one slide back. At the feature of fully automated systems, you see that there's three items basically reaching 100%, which is filter changer, vessel filling, and automated cleaning, which indeed, to our opinion, are the key features that define a fully automated system. Because if you don't have those, it's impossible to have multiple runs done consecutively in an automated way. Then again, the trend for online is a bit the same. Online UV, we see close to 80%, and even close to 40% of responders use online LC. Of course, with a fully automated system, you can argue the benefit of online measurements is even larger, because now you can not only do, let's say, one run in the evening when you go home, but you can do full continuous runs with analysis at the same time. So when you come back in your lab after the night or after the weekend, you would have the results of all those runs ran over that period already available. Another feature we asked for is whether your system is capable to prepare media from concentrated medium sources. And there we also see that around 70% of the people indeed have that capability in their systems. So that means you would prepare a smaller volume of a more concentrated medium, and then your system would prepare the final medium from that by diluting it with water. The advantage of that being you need to prepare less volumes, and it also takes up less space, because you can imagine if you're running multiple dissolution runs consecutively, you will need a lot of medium next to your automated dissolution tester. Another feature is then this orange bar, the option for automated basket testing. And that's indeed an interesting one, because automating a paddle method is still, let's say, more straightforward. You drop in the tablet and you start stirring. At the end, you clean the vessel and you're done. For a basket method, of course, you have to actually attach the basket to the shaft and then retrieve it after the run to then do the same for the next run. So it's a bit of a more complex feature, but still we see that a majority, around 80% of the systems has this capability. A similar comment for actually capsule or tablet testing with sinkers. You can, of course, preload your tablets or capsules in sinkers, and then a tablet rubber can drop them. But at the end of the run, you also have to retrieve the sinker. And there we see also around 70% of systems has that capability. And then the smallest bar you see there, what is it, brown, is actually the capability to do automated cannula filter changes. As Johanna also mentioned, this is less straightforward. We also see that a minor amount of the systems has this capability. So if that's something in your method you need, then, well, either you won't be able to use all systems, or you might need to look into whether your cannula filter can stay in place for multiple runs without causing any other issues down the line. Then you can move to the next slide, please. Another item we asked for is what type of sampling pumps the system has. In automation, the pumping of the sample is one of the key things, and there's three major types of pumps that you can find. It's peristaltic pumps, piston pumps, and syringe pumps. And looking at the top graph, we see that for semi-automated systems, the majority of systems seems to have piston and syringe pumps, but still some part of the systems also have peristaltic pumps. You'll also notice that this doesn't make up 100% in total. That's because responders could have multiple systems with different types of pumps, or maybe there's systems with multiple pumps within one system. And then if you look at the fully automated systems, there is more, let's say, the piston pump is dominating, and most systems do have a piston pump still, some minority with peristaltic and syringe pumps. Also important to consider here potentially is that people respond with the systems they currently have. Some of those systems might be very new, other systems might already be a bit older. And then we also ask to, given there's different types of pumps, whether people have any actual preference for a certain type of pump, and that's displayed in the bottom graph. And there you see for semi-automated systems, the preference more or less aligns with the prevalence of the type of pump. So mostly piston and syringe pumps are preferred over peristaltic pumps. For the fully automated systems, the piston pumps are the clear preferred one. And we also ask people to kind of motivate their preference, which I try to summarize here. So for peristaltic pumps, there's multiple drawbacks. They're not accurate, or not as accurate as the other ones, less reproducible. But probably the main one, I would say, is they lack power. So if you look at filtration step, if you want to filter through a syringe filter, that might not work with a peristaltic pump. The upside, on the other hand, is that they are quite easy to maintain because there's no direct contact of your medium with your pump. It's just a tube actually in the peristaltic pump. And that tube is also quite easy to replace, probably. Then you have the piston pumps. They are more accurate, more robust, more powerful, and they also can handle a higher flow. And I guess those two, how powerful they are and the higher flow, are probably the main advantages in my personal opinion. And they're also relatively maintenance-free. The downside is they're more complex. While a peristatic pump, if something is wrong, you can probably troubleshoot it quite easily by yourself. That's mostly not the case for piston pumps. And then when we look at syringe pumps, there the upsides are this. They are probably the most accurate. They're a bit simpler than a piston pump. And this is also an item that will come back later in the main struggles. A feedback that was given is that syringe pumps lead to less empty files. There's not really downsides listed here, but coming back to the piston, I would say probably a syringe pump cannot reach the same high flows as a piston pump can. So that would be probably a drawback of a syringe pump. And then you can move to the next slide, please. Then we also asked to software use, also discussed in the introduction. We see more and more software. So we asked people how they actually operate their semi or fully automated systems. So we see that for semi-automated systems, there's still a minority, around 20% of people operate the system purely through firmware, but the majority is already using the vendor software. And for the fully automated system, that balance is shifted even more towards running it through the software. And that's a general trend, I think, that we see more and more software driven. There is upsides to it, of course. It allows for better and easier data and user management. And it's often also easier to operate more complex systems. And that's why probably we see also that trend for fully automated systems. There's also some downsides to it. The addition of software complexifies the system qualification, as mentioned before. The added value of having the software does not always relate to the added complexity, and sometimes the software is not specific for a certain system and contains a lot of features that you might not need specifically, but they are still there. And then if you have multiple techniques and technologies across your lab, you might also have a multiplication of software applications that are vendor specific, leading to a very complex, let's say, software landscape. You can move to the next slide. So now we shift gears a bit. We discussed the features of the automated systems, and then we also asked some questions on actually how people develop methods on their automated systems, and we zoomed in on a few specific items. The first is the prime and waste drop volume. So to repeat, the prime volume is the volume that you pump before a sample is taken to actually prime the autosampler tubing with your current sample. And what we asked is whether people develop the prime volume specifically for a certain product, or whether they use a generic setting for that. And then you see on the top graph that for both semi and fully automated systems, it's kind of a 50-50 mix, I would say, where some people use a generic setting while some develop it product specifically. And then we also asked how you then validate the suitability of that prime volume, whether it's done during method validation as an equivalent study, whether no study is done or any other. So you see the majority actually does a method validation or equivalent study. When we looked at the detailed comments for people responding other, you can also kind of refer to it as a sort of equivalence test. And then, my interpretation of the respondents saying no study is that probably they would use generic settings, and they have also a generic validation or statement why that volume is suitable. The next slide, please. So we asked exactly the same for the waste drop volume. So again, the waste drop volume is the volume you would discard to the waste before sample collection. And so you'd first pump the prime volume, then you would pump the waste drop volume to the waste, and then you take your actual sample. Here we see there is more a trend towards generic value for this setting, and the minority is doing it product specific. And also for the validation of that, we see it's kind of a mix of method validation or equivalence, because again, here people responding other would be kind of a combination or still one of both validation or equivalence. Then you can move to the next slide, please. And that's also an important topic for automated systems is the cleaning, because with indeed having all those tubings in a semi-automated and fully automated systems, cleaning becomes ever more important. So here we asked how people clean their systems. Do you use pure water? Do you have a generic cleaning procedure? Do you have a product-specific cleaning procedure or other? So you see that it's mostly a mix, both for semi and fully automated systems, between generic and product-specific cleaning methods. The responders replying other, if you look at the comments, it's mostly a stepwise approach where they would have a generic methodology and they would check whether that works. If that works, they stick to the generic one. If that doesn't work, they would, for that product, develop a specific one. Then we also asked how people then actually validate their cleaning methodology. Do you do it during development, or do you really do an official validation for it? And do you use in-house criteria you set yourself, or do you use the same criteria you use for specificity, or anything else? And we see in majority of cases, it's actually a development study with either or in-house or specificity criteria, and only a minority, I would say 20 to 30% between semi and fully automated systems, is it part of the actual method validation. Finally, we also asked if you then test the effectiveness of your cleaning method, what do you actually check for? And there you can clearly see majority is really checking for API only. We asked, do you check API, formulation excipients, or media constituents? We see the majority checks API only. Then when we looked into the other answers, the majority there would test API plus media constituents, and only a very few responders, I think, also check for carryover of excipients. It might also be specific cases. We don't have those details, obviously. And then the next slide, please. So finally, for method development, we also asked how people are proving equivalence between automated systems and how method transfers are handled. So in the top graph, you see how equivalence between automated systems of different brands, for example, are shown, whether it's a development study or during validation, and again, what kind of criteria are used. We see this is really almost a fair mix. So of development study with in-house criteria, a validation study with in-house criteria, validation study with intermediate precision criteria, or the other. And in other, we saw it's actually also kind of an equivalent study, let's say, with certain criteria. And in the in-house criteria, we see, for example, that could be an F2 test or something in similar to that. For method transfer, we actually asked if you have automated the method, do you then transfer the manual method only, or do you transfer the automated method, or do you take any other approach? So we see that roughly 50% of responders transfer the manual method only. So then you could assume that if the receiving lab has any automation, they would have to show the suitability of that automated system in their own lab. Then let's say 30 to 40%, they actually transfer the automated methods. And then there is still a minor amount of people replying other. If we looked in the details, you could also interpret it as a sort of method transfer of the automated method. So that concludes the questions on the method development. And then finally, you can move to the next slide, please. We asked through different questions what the most common issues and hurdles were. And here I try to summarize them. I also try to order them in order of prevalence, just to get an idea what really the most common ones are. So if you look at the top one, we see that indeed sampling malfunction or poorly filled empty files is one of the most common issues. This could also explain why so many people actually have online UV or capabilities, because then you actually could skip the need to fill a file at all. Secondly, we see also a lot of temperature out of range, specifically when lower volumes are used. For example, if you use a 500 milliliter dissolution volume, and that can probably be attributed to the fact that, yes, these automated systems have to pump quite some medium around with the prime volumes being pumped first. That medium is then no longer exposed to the heating source in the vessel and could cool off. And then when it comes back to the vessel, it might lower the temperature of your vessel. A filter changer blocking is also quite a common issue. So any issues with that filter changer, this might also well be a reason why some people still opt not to have a filter changer and take other approaches. Again, here we come back to software issues, qualifications or limitations. So really the software either giving faulty errors, for example, or making your qualification complex, or even limiting you in what you can actually do with the system. Inconsistent sample introduction also comes back. So sample not dropping, for example, or dropping in the wrong position. We see fails of the online UV, and that's of course then one of the main downsides of online testing. If you're not filling files, if your analysis fails, you will have to redo your dissolution. While if you are classically sampling in files, and then your analysis fails, you might be able to reuse the same samples and reanalyze them, and not necessarily have to repeat the actual dissolution tests. The equivalence to manual testing in between automated systems was also listed as one of the common hurdles. The lack of suitable filters, so the right pore sizes not being available, or the required filters giving too much back pressure leading to breaking of filters. Foaming of surfactant media when pumping is also listed as a common issue. The connections of tubings, there's many tubings often connected in multiple places to pumps, to the bath, to maybe a filter station. Those connections actually failing or wearing out. And then finally, carryover was also listed. And that links back to that cleaning methodology you use and how you actually validate it.
Thank you. So we are at time. So with that, I will thank you, Nathan, Johanna, and Amjad for the insightful discussions. And also thank you from the member from the IQ team for supporting this webinar. And also I would appreciate all the people who joined this for the active participation and the thoughtful questions. And with that, I will conclude this webinar, and we hope you find it valuable and informative. With that, have a great day, and look forward to seeing you in the future sessions. Thank you.
Thank you, everyone.
Thank you. Bye.
Bye.